Hello everyone, welcome back to my channel StudyWise, and today we're going to be diving into the Cambridge International A-Level Biology, also known as the 9700 syllabus, specifically focusing on paper 5. This is a crucial part of the exam that tests your practical skills, so let's get into it. This paper tests your ability to plan experiments, analyze data, and evaluate methods and results. It's crucial to understand the format and what the examiner is basically looking for. So before we start, make sure that you subscribe, like, and leave a comment and turn on the notification bell so that you get notified whenever I post another video of paper 5. This is gonna be a series, so I'll be creating a playlist of explaining further details about each subtopic. This video is basically intro so that you get familiar with the how the questions are divided in the paper and so on. So before we start, you need to know that this paper is basically planning, analysis, and evaluation. The duration of this paper is 1 hour and 15 minutes. And it's 30 marks. And what could actually seem quite surprising is that it only holds 11.5% of your full A-level grade. So worst case scenario that you mess up in this paper, it's not gonna be a big of a deal because you have other papers that would carry you, right? But then you need to also score well in this one. So this paper is gonna be basically divided into two questions. Question 1 and question 2. Sometimes in some unique cases there's gonna be an extra question, but that's not gonna be an issue. So basically question 1 is gonna be mostly focusing on planning. And question 2 would have more stat questions, which is basically statistics. So I'll start with explaining what could possibly come in stats, and then I'll go back to question 1. So there's some basic calculations and stuff that you need to know, and then there's the stuff that you'll learn in bio. So the general things that you need to know, such as how to calculate mean, which is the sum of all the numbers you have over the total numbers of objects. For example, you have the length of something, which is 2, 3, 4, 5, and 6. You're gonna add these all and then divide them by the number of numbers, which is 1, 2, 3, 4, 5. 5. And then you have median, which is basically the middle number, so you need to arrange the numbers from the least or the lowest to the largest, and then you would find the middle number. For example, you have 1, 2, 3, 4, 5, 6, 7, and then you would start cancelling and find the middle number. And then you have mode, and then there's 3 types of mode. It's either unimodal, bimodal, or multimodal. So basically mode is if you have a set of numbers and then there's a number that is continuously repeated. For example, 1, 2, 2, 2, 2, 3, 4, 5. So the 2 is the most repeated, right? So it's gonna be a unimodal. In case you have a 1, 2, 2, 4, 4, 5, 6, 7, the 2's and the 4's are equally repeated, right? And then the 2's and 4's are the most repeated from the sequence of number. So this is gonna be a bimodal, and so on. And of course you have the range. It's basically the lowest to the highest. And if they ask you for the range value, it's gonna be the highest minus the lowest. So basically you're gonna have to find the difference. And then you have the IQR, which is the middle 50%. And I'll be explaining that also in the following videos in my playlist. And the stuff that you really need to be like well familiar with in paper 5 is 1. Standard deviation. Basically this shows like where the spread of data above and below the mean. And this formula you really don't need to like memorize it, but you need to know how to use it. Where n is the number of replicates, or basically like the number of samples. And this is the mean. Alright, so 2 is gonna be the standard error. Which is basically equal to standard deviation over the square root of the number of samples into the number of replicates, of course. And it shows like the error that lies in a sample mean. 3 is that you need to know how to draw error bars. And then about the statistical tests, there are t-tests, chi-square, and the formulas are also not needed to be memorized, but here they are. The mean of the sample 1 over the mean of sample 2. Basically you're comparing between two means over the square root of standard error 1 squared plus number of samples of sample 1 and the number of samples of sample 2. And then you have chi-square. It's basically comparing between expected and observed. It's equal to the sum of observed minus expected squared over expected. And then you have the correlation tests, which are basically Pearson and Spearman. And these are to show like if two samples are like linearly correlated. And then you have the Hardy-Weinberg. And this is like very, very crucial for both your paper 5 and your paper 4s, because like it comes with like 4 marks. And it's like very, very easy. So why lose that? Anyways, and then there's a formula, and then there's like a set of conditions. We're also gonna be discussing that later. And then for biodiversity, you need to know the Simpson index of diversity. Which is 1 minus the sum of n over n total square. And then for the sampling techniques, there's like many others, but specifically like the percentage covered, the frequency, the density. We're going to be explaining that in biodiversity and the types of sampling and so on. That's basically like in question 1. And you need to know for like the mark and release recapture method. Mark, release, recapture method. You need to know this formula, which is the first number like the total number of sample you took in the first capture multiplied into the second number of samples over M2, which is the number of marked organisms in the second sample. This is very important, and you do need to memorize this one specifically. So basically this summarizes like the things you need to know in the question 2, basically statistics, and all of these are going to be like explained in the playlist. You also need to know like how to calculate the standard deviation using a calculator, which is very very important because like it saves time and you could ensure that you correctly solved it. Alright, so now we're going to be diving into question 1. This question basically tests your practical skills and planning skills, of course, and is usually the most extensive question on the paper, carrying the most marks. Here's what you need to do to tackle. Of course, I'll be like posting specific videos on explaining specific techniques and biological experiments that you're supposed to be like familiar with. And also I'll solve a few of the other questions in the past papers and give tips and tricks. So make sure that you turn on the notification bell to stay tuned. Alright, so there's specific ways that you need to know so that you could tackle it quickly and basically understand the question. So first of all, you need to read the question carefully because you're often given a scenario involving a biological experiment. So you need to look for the hypothesis, which will basically give you a guide of the rest of your planning. So you'd either be asked for identification or method. The method is basically like 8 marks. 8. It could reach to like 12, but you'd be lucky if it did. And then identification. So both of them you go through the same like scenario of answering it, or like the same method. So you need to start with like the hypothesis. Hypothesis is basically the aim of the experiment. For example, if we're gonna be carrying an experiment about photosynthesis, right? So the hypothesis, and then we want to like check the effect of increasing the power of the plant, or like increasing light intensity. So the hypothesis, for example, would be like increasing light intensity increases the rate of photosynthesis. So you'll be asked about the independent. What is the independent? Independent is the variable that you're changing, like the variable that you're seeing the effect of it. So here it's going to be the light intensity. So there's several ways of changing light intensity. For example, let's say we're going to be changing the power. So we'll say change the light intensity by changing the power of the lamp, and then you need to set like 5 values. For example, like 10, 20, 30, 40, 50 watts, and so on. And then you have to state the dependent, which is what you're measuring. For example, its rate of photosynthesis. They usually give clues for it in the question, but for example, let's say the volume of oxygen gas produced using a syringe. So you're going to say measure the volume of the oxygen gas produced using a syringe. And then you need to say a time duration, right? For example, during 1 hour using a stopwatch, and so on. And then variables to be standardized. for example, you need to standardize the amount of carbon dioxide it's consuming, right? So you're gonna do that by sodium hydrogen carbonate. And then, like, this is what you're gonna be standardizing. You need to say how are you gonna be standardizing it. For example, add 3 grams, or like you could use it as like a volume or like grams. 3 grams of NaHCO3, and then you need to say the percentage. So for example, 0.02%. And then if it's grams, you're gonna be using a digital balance. If it's a liquid, for example, like 3 centimeter cube, let's assume that, you're gonna be using like a graduated pipette, and so on. And of course, like the temperature, the humidity using a dehumidifier, and so on. And then you need to say the reliability. So basically, that's like how, like the replicates. For example, I'm gonna replicate the experiment like 7 times. I'll exclude the anomalous. I'll calculate the mean of the dependent variable. So you need to say that because it comes with like 2 marks, which is very, very important. For example, like here we're investigating the effect of changing the light intensity. So we'll say replicate the experiment 7 times for each light intensity or power, then exclude anomalous, and calculate mean rate of photosynthesis. And then you need to say the risk. First of all, you need to say whether it's like a high risk, medium risk, or a low risk. For example, let's assume that we're using like, okay, we're using NaHCO3, sodium hydrogen carbonate, which is an irritant, right? So you're gonna say, for example, it's a medium risk. And then, what is the risk? NaHCO3 is an irritant. Like, it irritates the skin. And then you're gonna say, like, how are we gonna prevent this risk? For example, wear gloves. If you're gonna cut something, use a cutting board, avoid cutting yourself, it's a medium risk, and so on. So it's nothing basically to like memorize, but you have to get yourself familiar with like a specific set of experiments, like biological experiments, for example, like the plant related, such as photosynthesis, uh, osmosis, diffusion, the transpiration. Like this is basically like a photometer. What else? Like the immobilization of algae, the plant disc experiments, like where you have like you cut like discs out of a leaf lamella, and then you're supposed to calculate the time taken or the distance or so on about like the disc moving from the bottom to the surface, indicating the rate of photosynthesis. Chromatography. Like there are a ton of other experiments like enzymes. For example, like they could ask you like the effect of caffeine, or like basically like something that affects your reaction time. And several others. Like there are a ton of others. If you say biodiversity, we're talking about sampling. So there's like random sampling. So it could be random, it could be systematic, and then you could like, it could be a mark release or capture. You could use the systematic or like the random and then calculate the percentage cover. There are many of like specific sets of experiments that are usually repeated in many exams, so you need to get yourself familiar with, and of course I'm gonna explain all that in separate videos in details. They're not gonna be like more than 10 minutes each, so they're gonna be good for like cramming before an exam. So I think we're done with a pretty like good explanation or like a brief for question 1 and 2. So thank you, and please make sure that you subscribe, like, and leave a comment, and turn on the notification bell to stay tuned for following videos. Thank you.